down to your clinic was probably like the final straw that led me to start this show. Because along the way, while I was building my company, I had to search for solutions for my own infection problems. I found an API overseas that I reformulated and ended up getting FDA cleared. Then I turned it into a product and built the company. And I kept experiencing people, innovators, entrepreneurs that had these incredible ideas that they just couldn't get here, either because of antiquated regulatory frameworks or because I almost want to say the traditional medical guard wouldn't accept it, as a potential new manner of care.
Mm-hmm.
But nothing hit me harder than when I brought Luca down for his inflammation to your clinic. And it was my wife, I talked about her on another episode, that went on like full Sherlock Holmes mode, looking for things to help Luca. And she found stem cells. And of course, I didn't know a ton about stem cells. I actually interestingly knew about regenerative medicine when it comes to wound care, because I'm in the wound care space and I've actually, I've created a product, it's a collagen product for wounds. Today's guest, Dr. Riordan, I knew him before he knew me because I'm a pharma guy, which means nothing was going near my kid without me understanding everything about it. The quality systems, the production process, the science behind it, the number of patients. And I actually called a number of clinics, and I'm sure you've heard this story before, Dr. Riordan, and everybody says, our cells are clean, our cells are from here, our cells are from Europe, our cells are better. Nobody has touched as many patients as you. I would inarguably call you probably the grandfather of this science. But if I think of, I know this morning I was driving down here and I thought of this, like the Sistine Chapel, there's that painting of that thing that's just out of reach, like the hand reaching out with the finger. And I think about your clinic in South America, and it is the thing that I have seen thus far that I most want everybody to have. So with that, I'd love for you to talk about your work and introduce yourself.
Well, thanks, Bradley. I've been, I guess, a researcher, and I worked at the cancer research lab for 14 years. And then I had a patient that we treated with something that we had developed there. And she had a stage 4 cancer. And it was the same stage 4 cancer that my best friend's mom had. And I watched her pass away at the age of 18 and felt completely helpless. Well, that was, I don't know, in the 1970s. And it was just, they treated her like dead woman walking and didn't give her any care. I remember going to visit her in the hospital and she was completely disrespected. And she died, cachexia and basically malnutrition in the hospital. And there was just nothing I could do. And that stuck with me. And then we had a lady in a clinical trial that we were doing with dendritic cells. This was in Kansas at my father's clinic. Lady's name was Esther Campbell. She had the exact same disease, and but it was stage 4, but it wasn't terrible like Esther's. And her husband's a family practice doc, and we got a compassionate use exemption for her to treat her with this, basically training the immune system and with dendritic cells, training her immune system to recognize and destroy the cancer. And she did that therapy, and in 6 months she was tumor free. And I'm in Kansas, and the medical board's not going to let me do that. So I looked at places in the United States that had either right to try laws, or it was called something else back then, but
Compassionate use, I think, was the term.
yeah, it was just basically the terminal case. But there were only 2 states. There was Alaska and Georgia that had those medical freedom. It's kind of medical freedom act, and then and Arizona had some different rules. They had a naturopathic medical board and a homeopathic medical board and a regular medical board. And I went to both of those boards, and I told them exactly what I wanted to do. I wanted to do high dose intravenous vitamin C, which my father and I had shown that you can achieve blood levels of that kill tumor cells and leave normal cells alone. And I also wanted to do the dendritic cell vaccine just on a patient by patient basis. And both medical boards in Arizona said absolutely. So I moved to Arizona, quit my job, moved my wife and 4 kids to Arizona in 1999. And I started a clinic there, the Aiden Clinic. And we made did root cell vaccines. It was quite successful. And then my wife wanted to move to England. She was English, and after 3 years of that business, we moved to England, and for the kids to get exposure over there. And so I had to sell that clinic. And when I did, I had a non-compete in the U.S. I had been doing vaccine work with a clinic, immune therapy clinic in the Bahamas, and I wound up buying that clinic in 2003. And I already had a laboratory established there, and I went up buying majority shareholdership of that company. So when I got back from that year sabbatical in England, we moved to the Bahamas and stuck in there, and that's really where I got started with with mesenchymal stem cells. My first patent application in the what I call the cell therapy world was on a way to expand the one true stem cell you have as an adult, which is the HSC. That's the stem cell that creates all of your blood cells, your white blood cells, your red cells, and your platelets. And it predominantly resides in the bone marrow. But those things, you can isolate them. And I needed a product from those cells. I needed those to divide to make more of a cell that I was going to use for my vaccine. Several years in the laboratory, we were investigating different things that could stimulate those HSCs, hematopoietic stem cells, to divide outside of the body. And the one thing that I found worked better than anything else was slices of placenta. So these slices of placenta would secrete factors unknown to us at the time that would stimulate, would allow for these cells to divide in culture. Otherwise, they just don't like to do it. So that's how I got involved in, and of course, when I was doing cancer research, we had 60 tumor cell lines growing. So one thing I learned well is how to grow cells outside of the body. That's the background on how I got started with MSCs. And then we did our first MSC treatments in the Bahamas.
What is a stem cell? And then we'll get into the different types of stem cells and how you landed on the MSCs as your choice.
MSC is called a stem cell. It does not behave like a stem cell in the human body, which is hard pill for a lot of people to swallow. Your body needs certain cells that can protect you from infection and can carry oxygen to your body, take it from your lungs to the rest of your body, get rid of waste products in your lungs like CO2. And those are all produced by the stem cell called the HSC. It's in your bone marrow, and it divides and makes all these wonderful cells that keep you healthy and help you repair your body. And then each one of your organs has another kind of stem cell that's specific. We have stem cells in your skin, in your liver, your kidney, your heart, your lungs, your intestines. They all have tissue-specific stem cells. And right next to every one of those tissue-specific stem cells sits an MSC. So the MSC provides nutrients and direction to the organ so that it can repair itself. And it also provides energy to your body by donating little parts of a cell to cells that are in trouble, called the mitochondria. So it provides energy. And they're found all throughout your body. They're found in every tissue in your body. For example, when a baby's born, the percentage of cells in the bone marrow that are MSCs is very, very large. And then you have to go from a 7-pound baby to a full-grown adult, and these cells distribute throughout the body. And so the number of the bone marrow goes down, and you have fewer and fewer of them. And if you look over time, by the time we're my age, the number is distressingly low, and it continues to decrease with age. So that's the normal course of events. You can do things that can help maintain your MSC population. But the only two true stem cells in your body, the only true one, in my opinion, is the hematopoietic stem cell. because it supplies cells to the entire body. And then you have the tissue-specific ones, which are true stem cells, but they're committed to a particular tissue or an organ.
So the hematopoietic stem cells, are those the ones that can divide and become anything?
Well, we have to back up because the ones that can divide and become anything are embryonic stem cells. Because think about it, you got to make a baby. So the egg is fertilized, and that the first division, second division, up to several divisions, all of those cells that are there are not fully committed to becoming anything. Those are embryonic stem cells. And there are a couple reasons that I have not pursued working with embryonic stem cells over the years. And the first of which is I don't like to mess with Mother Nature. And what happens, the way you identify embryonic stem cell is inject them into an animal and it forms a teratoma, which is a sort of a ball of tissue that includes hair and teeth and skin and all kinds of lovely stuff. But it forms a tumor, a non-malignant tumor. But that's the number one reason I never wanted to work with embryonic stem cells. The second is that in order to grow them from that state long enough to in culture to get them to become something you want them to become, there are all kinds of damages that occur to the fuel producing units of the cell, which are called mitochondria. And there's all kinds of genetic things that happen. So then the proteins that the mitochondria are producing are now so different than what they were, from an earlier state, that the immune system rejects them. So embryonic stem cells are problematic for two main reasons. One is they can form tumors, big problem. Number two, they become antigenic to the recipient. So even if it's the same, you're doing it to the same person. So now they just look different. So we can do this thing called induced pluripotent stem cells, which is taking an old cell and reversing it either genetically or with chemicals to become like an embryonic stem cell. When you grow those up to large enough numbers for them to be useful, they are antigenic to the person that they came from.
So over time, and again, I cheated because I read your book and I liked it, by the way. You've taken care of, treated, your clinic has used these MSCs in tens of thousands of people, right?
I think our latest number is 17,000 people in our clinic. And according to estimates that Ari and I garnered years ago, it was, I think globally, it's probably over 100,000 people have received them, maybe even more. It's kind of hard because not every country reports everything that they're doing, but I don't know the exact number. Last year we treated 2,500 people.
I can tell you when I showed up in the airport down there, there were 50 people showing up that day for a 5-day course. I don't like that I have to fly down there, and I think that you would spend every last penny you had so that people didn't have to, based on what I've gotten to know about you. But a lot of people do, and that's what bothers me, and I know bothers you, that they are not accessible to everybody. So from what I recall you telling me and your book saying is that over the course of treating patients, you saw, everybody seemed to have, from what I read, a positive outcome, or the people were reporting positive outcomes for the most part. Some people were just having like stupendous outcomes, correct? And then you identified some particular markers that became, we'll just call them, Riordan's favorites.
Yeah, that was an interesting study we did. Yeah, we had some people that just had miraculous results. Not everybody got better in the early days. Most people improved, the majority enough to warrant going forward. But we did have cases that I would just consider miraculous. In one example, because it shows kind of how it went. We had a high school football coach from Texas, and he's been all over the news, and he doesn't mind me using his name, is Coach Sam Harrell, 3-time state champion football coach in Texas. Came to us, and we treated him, and he had a very, very strong response, and he vastly improved, and he was able to he was able to walk, ambulate without any assistance. And that lasted for about 14 months, as I recall. And he decided to come back to treatment. And the second time, he got some benefit, but it was nothing like we saw the first time. So he wound up coming back less than a year later, and we treated him, and the it was just miraculous what happened. It basically had no deficit 3 months afterwards, and that was a very prolonged response. And and so immediately you kind of go, well, what's different about 2012, 2011, 2010? What's the difference of those cells? And then I went to all the doctors and said, let's get all these cases where you have somebody that had maybe not a great response and then a great response. Somebody that just had a miraculous response. We huddled up and we got a number of these cases. And one thing I've always done is that never throw anything away, which drives some of my people crazy. I always save back every lot that we've ever used, because what if something happens? What if you want to test it and you use it all or you throw it away? We took these miracle cases, what I just call miracle cases, that highly clinically efficacious things that occurred. And we took the cells that were used for those. We took, say, the middle ground, which is like 2010, and then the lower ground, which is 2011, where it was not, he didn't have much of a response. We grew up these cell lots in large quantities so that we could then test them. And so we had, golden, silver, and the bronze, I would say. And then we grew them both in one layer and also in 3D culture, because I knew ultimately we were going to have to go to 3-dimensional culture, 2-dimensional culture. That's a little bit complicated, but I knew it was going to 3 dimensions, so I wanted to test in 3 dimensions as well. And then we looked at different markers from these different cell lots, and we found that these ones, that the highly efficacious cells, these golden cells, had markers that we could identify them early on in the culture process that were exceedingly different from both what I call the silver cells and the bronze cells. You do pharmaceutical, so you know what p-values are, highly significant. That is not chance, that these are true differences between these cells. At that point, we started screening all donations for, we would start a primary culture, and then we would screen them for these markers, and we would be able to determine whether or not they were going to be useful. And we wound up throwing away many, many cords. As another cord, why should we even use it? And so these are renamed them golden cells, for lack of a better term. And we've been using them ever since. And I think our results improved dramatically. One of the things we do is we have a patient registry of everybody we've ever treated. We follow them up. So we have an extraordinary amount of data on historical data on our patients, and we record every side effect. And it's an enormous database of people with different conditions have been treated over the years.
The other thing, if I recall, because I had a chance to visit your production site, which I swear if you had said it was a Merck or a Pfizer site, I would have believed you. I mean, this was one of the most rigorous quality control production setups I've ever seen, which, again, as a parent of somebody who received them, I appreciate very much. I'm not somebody who actually likes to go out there and talk negatively about anybody else doing something, but you probably have seen places creating, selling, infusing, injecting things that aren't as controlled. I mean, is that a fair statement?
That's a very fair statement. I'll just give you a little bit of what goes into our manufacturing process.
Yeah.
One of the things that we have to do on a cell lot before we release it is adventitial viral testing. And that costs, at the bare minimum per lot, that costs $250,000 to do.
That's testing them to make sure that they don't have viruses you're going to pass on to the person who receives them.
Right.
Yeah.
Right. It's a very rigorous multi-month process. We have the most stringent, and by the way, FDA audited us, and they spent 2 weeks there, and they were actually quite pleased at how well we document everything, how well we test all of our products, the testing on each lot. If I were not to do the things that FDA requires for a manufacturer to do in the United States, and I just were going to make cells and I would test them at the end, they would cost less than 10% of what it costs us to manufacture. To comply with FDA regulations, we need to essentially close our manufacturing twice a year for something called media fills. There are no regulations ex-US for media fills. There's no regulations for environmental monitoring in many parts of the world. Last year, we spent $770,000 just on the plates that we use for environmental monitoring to ensure that there's no possibility of anything growing or contaminating any of our products. We test 10% of our product, each lot, for sterility.
That's a big amount of a lot. Speaking as a pharmaceutical manufacturer, that's a big chunk.
It's one thing to do GMP. It's one thing to do GTP. I think the majority of the places out there are operating under what I consider GTP, good tissue practices. And, at least that's better than nothing. But to do full U.S. FDA complying with the latest regulations, which are quite old, by the way, their guidance to industry on cell and gene therapy, cell and tissue products, it's a very high bar and it's a very expensive bar.
So from what I recall, the Updated Modernization Act of 1997 was the last real reform that we had. And I know there's been some recommendations since then, toward being a little bit more accepting of regenerative therapies, for example. But, and I believe in the Cures Act that there were some additional recommendations. There were no mandates given. They were saying, we urge you to look at these more, but Congress didn't give FDA a new set of rules to actually regulate them by. But you're stuck with 40-year-old rules with, borderline 2050 futuristic technology. It's got to be unbelievably frustrating.
Yeah, it is. There are parts, I understand that they wrote those regs for the safety, and I appreciate the sentiment of trying to have a safe product. But at the same time, I think myself and other people who are in this industry that are doing it right could make a very compelling argument that some of the things that we're not forced to do, but if we don't do them, we get shut down. So you can call it whatever you want, that we have to do to be compliant with the latest regulations are complete overkill. Just as an example, we do these media fills to show that there's no way that anything bad you did in our product. And you basically are testing every input into the process, and that shuts your manufacturing down for at least a month, a year, and twice a year, by the way, 2 times, 6 months apart. And every time we do a manufacturing run, it's tested for sterility. 10% of the product is tested for sterility. So in my mind, and in the mind of other leaders in this space, every time we do a manufacturing run, it acts as a media fill. And so why do we increase the price of our product, the price of manufacturing by this tremendous amount to do what we believe is overkill and not required? Now, certainly if you were to have a contaminated lot, then that's the time to do media fill, but we don't ever have contaminated lots. So why are we shutting down our business 2 months a year? And not just shutting down our business, but we still have to spend. I still have to pay everybody, and also I have to pay for all the product that I'm just going to throw away at the end of the media bill. So it's kind of hard to fathom why we still have to do that.
Yeah, no, I call that a dress rehearsal for an event you already went to.
That's what it feels like.
Yeah, and an expensive dress rehearsal about like you got to buy all the clothes all over again each time. And then you think about those cells, and I know you well enough to know you'd rather donate them to people who need them than just throw them out.
That's a perfect way to put it. Yeah, dress rehearsal for something that's already happened, or that happens every week.
Panama, just to jump back to that.
Yeah.
They were open-armed and receptive is the vibe I get down there to you setting up an incredible clinic. Has that been your experience overall? Besides, it's really nice down there, by the way. It's kind of like Miami. It's a really beautiful place, but like, how was the reception there for, this opportunity for their population to have access to it?
We've been up and we've been down. I'll put it like that. And I can tell you, we've been there been fully functioning for 17 years. So we're doing something right. We've done 3 human clinical trials there, phase 1, phase 2A. I can't tell you how many times the tourism office has called me and said, "Hey, can we interview so-and-so because we know they're at your clinic?" Our clinic is attached to the Hilton Hotel, so you don't even need to go outside. You can just, as you well know, you were there, you just walk from the lobby and you get another key to our elevator, and you go to the 63rd floor, and then you're in the clinic. The reason I went to Panama to start with was I met Dr. Paz, who was a medical director.
I got to meet him first. He's such a nice guy. He actually looked over Luca's labs over WhatsApp for me because we had a mutual friend. He's just the kindest guy.
Yeah, I met Dr. Paz and Dr. Fernandez, who are laboratory director, and those guys, um, they wound up coming up to look at my other lab and said, "Hey, I think we can do this in Panama." And they got a meeting, and Rodolfo, Dr. Fernandez is good friends with the president, and so we had all a series of meetings, and we met with this what's called the City of Knowledge, which was our former the former Army base, the U.S. Army base in Panama that they had given back, and they started an NGO there to basically utilize that property for. They wanted to bring biotech, they wanted to bring educational. So there's several universities that are in there, United Nations in there, and they accepted us into that.
Which is an application basis too. I've actually looked at that. I mean, there's some major industries in there. Yeah.
So we were accepted in there, and we were the first biotech company, and we built out our manufacturing facility in there. And they actually used us as sort of like, look, this is what's capable here. Because these buildings were, they're built in the 1940s by the Corps of Engineers, some of them older than that. And our building was just concrete, and there were no windows, there were no doors, there was no AC. We started from scratch, and with a generator. It took us 2.5 years to build that manufacturing facility out. And so they used our place as a showcase to look, this is what's possible out here. And now all the buildings are full. And we completed ours in 2010. We had a small suite before that. And then now we have something on the order. We have 50,000 square feet on the main manufacturing floor there. We have other space, but just the main manufacturing floor is 50,000 square feet. The president at the time saw this as an opportunity to create a biotech industry in Panama, and sure as heck, that's exactly what's happened. We have 130 employees. They're all, the vast majority of it had no experience in this. So the government did 2 things for me. We will give you signature visa rights. So in other places, I had a very difficult time bringing in talent because I'm coming into a country with, at the time, 3.5 million people with no biotechnology experience. And they had started a few years earlier a biotechnology track at the University of Panama and a couple other universities. And so we had people coming out with nowhere for them to go. And so we hired a lot of those people, and we've done what we promised, which we were gonna train the population. So now almost every employee is Panamanian. We have some 2 long, actually just 1 left out of four Costa Ricans we brought. And the government of Panama was just so very nice to me and inviting and giving us that visa right to bring in people that they're not going to stay forever, but we need somebody to train these people that just haven't had the training. And also there was a law in the books that was, it came across in 2004. The spirit of the law was to prohibit embryonic stem cell research. And that, I think, had more of a religious base than anything else, and also allow for adult stem cell research to take place, which, even though these are cells that are isolated from afterbirth, so a live, healthy baby is delivered, the umbilical cord is tied off and eventually cut, and then the mom delivers the placenta. You have a live, healthy, happy birth. And the placenta then comes to our lab, and that's where we isolate the cells from the umbilical cord tissue. So there's no harm to mom. There's no harm to fetus. There's no harm to potential life. Whereas with embryonic stem cells, you're you're basically taking a potential life and removing it. And then fetuses, we've never touched a fetus. Only live, healthy afterbirth is what we use.
We talked about right to try and compassionate use in the U.S. So down there's a law in the books that says you can use adult stem cells. And just to reiterate what you just said, anything after fetal and umbilical cord forward is considered an adult stem cell.
Correct. After a person is born, everything after that point is considered adult.
In the US, if somebody has a condition, can't travel, for example, maybe it's something, maybe there's an ambulatory reason, a pulmonary reason, any reason of a number, wanted to get, and their physician thought it would be a good opportunity to use your stem cells on this patient. What's the process like for that patient, that one patient, to get your stem cells here?
There's a federal right to try law that was passed, I think, in 2018, and it allows for doctors to treat patients and for manufacturers that have investigational products, including drugs and biologics, to provide those to a patient who has a terminal illness. And that's called the Right to Try Act, the Federal Right to Try Act. And since that time, some states have expanded that right to try. And in fact, in Texas, there was a right to try law that went into effect September of 2023. It's very harmonious with the federal law in that it requires that the manufacturer of the cells has completed a phase 1 clinical trial, a safety trial, and that they also have ongoing clinical trials. And so because of that, we qualify there because we've done multiple phase 1 trials. We have ongoing clinical trials. And our current single trial that we have open is for the treatment of Duchenne muscular dystrophy. And we have finished our pre-IND meeting with FDA and submitted our full application for a phase 2B trial, which that just means you're always looking for safety, but you're also looking at outcomes.
I want to clarify for everybody out there, getting FDA's blessing for what's called an investigational new drug, that's what IND stands for the listeners. It's not just a piece of paper and an application. It is usually a multi-million-dollar process. I mean, you have to go through what's called the CMC process, which is where you show FDA, every machine you use, proof that you can produce the same batch over and over with the same level of safety, the same level of potency, because the patient deserves that kind of quality. We all agree with that. Based upon your own chosen quality system, you would do that on your own. Even after 17,000 patients in Panama, you still would have had to have an open IND, gone through the submission process with FDA, had the meetings, gotten their blessing to start a clinical trial, started the clinical trial itself, which is another many millions of dollars. I do clinical trials. I know they are not cheap. And only because you have an ongoing clinical trial, an open IND, and you've completed the many millions of dollars worth of the quality and the paperwork that goes into the IND package, so we call it with FDA, then that one patient on a case-by-case basis with a terminal illness can get the care.
Yes, very good summary. In the case of Texas, the right to try law of 2023 says almost identical. It's almost identical to the federal right to try, but they allow it not just for terminal illnesses, but for severe chronic debilitating illnesses. And that passed in 2023. So in Texas, it is legal for somebody with a severe chronic debilitating disease. So I use the example of heart failure, which in Panama, they've treated a lot of heart failure with good success. Why should somebody with heart failure have to go to Panama to receive a treatment that's been demonstrated to have success? And it, more importantly, been demonstrated to be safe in many, many trials.
So that could be done on a state-by-state basis using Texas's model, though.
That's correct. And it's my understanding there are 15 states that have already passed something, either that exact same law or something very similar to it. And, on top of that, you have different states that pass stem cell laws. And this is where it gets a bit worrying for me because, I was an expert witness on multiple cases of people who are quote unquote using stem cells, and it gave people infections.
Yeah.
And they just had no quality systems in place. And that's why this happened. And I worry more about somebody getting hurt than I do about overregulation. So I think safety needs to be there first, but also I think there should be an alternative regulatory path that other countries have already instituted, that once you demonstrate that you make a safe product and you demonstrated that to whomever's satisfaction in the government, then you should be able to actually use that. Somebody with a severe chronic illness should be able to use that product, in my opinion.
I agree with you.
And a doctor should be able to give it to them. Somebody who's suffering and potentially doesn't need to suffer, I think it's just a horrible system that we have that makes them go to Panama or around, somewhere around the world to get something that, by all rights, as an American, I should be able to receive in my hometown. I don't understand why I have to go out of the country, but we've had 60,000 or 70,000 infusions. We've had 0 serious adverse events.
Yeah, which is statistically in pharma unlikely.
We've had side effects.
But a serious adverse event, and you're doing infusions. These are not like, a topical something or another. This is IV. So
yeah.
So that's amazing.
We're super happy about that. And we wish the whole industry would be like that and be, take every precaution, do no harm first. And I was listening to Huberman the other day, and there's a guy on there from Stanford. I haven't had a chance to look him up, but he's a stem cell researcher. He and Huberman were talking about these terrible things that have happened. There was one guy with a tumor. Well, it was an MS patient many, many years ago who had been to 3 or 4 clinics. He hadn't been to our clinic, thankfully, but he had been to Ukraine. He had been to Mexico, and he went somewhere else. I think actually, and he wound up having a non-malignant mass on his spinal cord because they injected these cells in his spinal cord. What I think is he got some fetal cells, or maybe even earlier than just mid-fetus, and those things just wanted to become a baby, so they grew into a mass.
Now,
so that's a horrible thing, right? That's one case. And then in Florida, there were 2 ladies that were treated for, eye conditions at a clinic, and they used their fat to isolate not only MSCs that are found in the fat, but the other cells in the fat, which are immune cells and, T regulatory cells and endothelial precursor cells. It's a gamish of cells, but predominant cell type is the MSC. And they injected, listen to this probably, they injected 2 ladies on the same day in both their eyes. First of all, with a product that's never been tested before.
Yeah.
That could not have had any serious quality control done on it because it's done on the day, same day procedure.
It's impossible.
So who knows what was in that product? Who knows how it was isolated? But it is not a typical stem cell product. Also, that is just poor judgment, poor medical decisions, malpractice. If you're going to do something first in man, and you have a willing participant, and it's IRB approved, and you're going to do it, well, no IRB would say, oh, do it in 2 ladies in the same day, or do it 2 people in the same day, much less do it in both eyes in the same day. You have 2 eyes. To use that as one of the examples, it's apples and oranges to compare to what we do. And then you look at the only other one I remember specifically was a lady who had, I think, fat-derived stem cells, and a plastic surgeon put them under her skin to plump up her, basically, I think, around her crow's feet on her eyes or something.
Yeah.
And he also used bone morphogenic protein. Who knows why he put that in there, but that was in there. So these cells actually became bones, little tiny bones that are fake.
Oh, come on. Seriously?
I'm serious. So somehow those, what I would call totally irresponsible, they're total malpractice, are used as examples of how stem cells harm people.
So the way I understand it at a very rudimentary level, what the MSCs do once you infuse them is, number one, they seem to seek out inflammation. And then they sort of park themselves where the inflammation is. I'm sure they park themselves all over, but, and then they obviously replicate. And because they're younger, even though they're still considered adult, they're younger. They're not as old as you or I, so they're going to replicate faster. And then it's not that they turn into anything like a fetal cell would, which can be dangerous because you don't want it turning into a tumor or a toe or something, and in the wrong place. Unless you really need a toe, of course, but that's a different story. So the way I understand it is that these MSCs park themselves in a theoretically inflamed spot, and then they start secreting like a magic juice, the secretome, I think you call it, the secretions, the trophic factors, the exosomes, all of those wonderful things. And it's a signaling mechanism that sort of allows or signals the body to heal itself.
I agree with everything you said. Um, think about how your body naturally repairs itself, and when does it repair itself? Why does it start repairing itself? If there was a constant on signal, um, then all your resources be exhausted pretty quickly. So the majority of your MSCs are in a quiescent state. They're found at the junction of every capillary, like I said earlier, and so they're found throughout your body. So imagine you get into a knife fight, you get cut. Well, what happens to the capillaries? They break. I mean, they're sliced through, and the resting MSC becomes activated because now it's released from a capillary, and it senses there's a problem, and then it starts secreting things that are going to stimulate other cells. It recruits other cells to the site, and it stimulates regeneration. And early on, there's a lot of inflammation because the immune system rushes in there because you're more likely to die of an infection than anything else at that point. So the immune system rushes in. The immune system, in that, as it's gobbling up bacteria that could possibly infect you and kill you, or other microbes, it's very inflammatory because that it has to do that. And that early in wound healing, you have this inflammatory phase where the immune system is dominant. And then the MSCs are able to divide and start secreting things that stimulate, that tell the immune system, like, hey, okay, you've done your job. Thank you. We got this. And they're secreting things that give energy to the surrounding cells. They can secrete their mitochondria that can be taken up by the injured cells around them. They secrete growth factors that stimulate those cells to grow. They produce things that prevent cells from undergoing programmed cell death. They secrete things that induce new blood vessels to grow. Because you just sliced through the blood vessel, now we have to have more blood vessels. They're just profoundly anti-inflammatory when they become activated, and they're profoundly immune modulating.
I know that you have two production facilities. You've got the one in Panama that services the Panama clinic, and you've got one in Texas that I've seen that is, I mean, there's got to be at least 40 or 50 people working there, multiple clean rooms, equipment that is like nerd Disneyland, inside of that place. That site is, and you're producing cells in there a lot. That site is just for research. I mean, you are spending a ton of money trying to get these cells to more people, through the FDA process.
Yeah, and we hope to get there eventually. And that we've set that up. We've spent the money. We've had significant investment. Tens of millions of dollars have been invested in that facility, with the hope that we'll have a robust manufacturing system that is completely closed, completely roboticized, and completely scalable. One of the challenges I see is once you get approval, who's going to make it? Because currently there's no facility that can make enough even for a relatively small condition. We're thinking 5-10 years ahead and trying to get ahead of it.
So one of the things that's always bothered me is doing things the right way, and which is harder, more expensive, takes longer. It's the war versus the battle. And then seeing people make the quick buck doing it the wrong way. But in the case of some of these, I'm going to use the term bad actor again, sort of fly by night shops out there that are just doing things without the level of quality control that you have. it scares the shit out of me. I heard a story about somebody who was, buying, like a peptide or something, one of these style things, an injectable peptide, calling it regenerative medicine from a hairdresser, and injecting herself, and ended up getting heavy metal poisoning. And they've become buzzwords almost, exosomes and peptides and this and that. And with that comes a demand, and with that comes people who see an opportunity to make a quick buck. And with that comes a huge amount of risk. So that scares me. And I know you well enough to know that you'll never stop until everybody has access to this, which is,
That's what I try.
yeah, that's why I started my business too, because I felt like I had something that could help people. And I felt a moral obligation, almost like a mandate to get it to them. Actually, I have an anecdote. I remember a good friend of mine who's an infectious disease specialist. He's actually a polio survivor. He's an amazing guy. His name's Jeff Galpin. When I started doing my original testing back in the day, this was just the in vitro testing and the human repeat insult patch testing and the basic biocompatibility stuff. And it turned out that my product that I had invented was really helpful, really gentle, really powerful, could really help in the case of, chronic wounds, diabetic ulcers, pre-amputation, stage wounds. I said, if you have something that can help people, and you don't do everything in your power to get it to them, pardon my language, I said, are you an asshole? He looked me right in the eye. He said, yes. And I love Dr. Galpin. He's the best. And it's exactly how I felt. I mean, so I know that, you have a very well functioning business down there, and I value and appreciate and have so much gratitude for the fact that you're not stopping until everybody has it. The model of that sort of miracle that is just out of reach here, at least geographically. You need a plane ticket still to get. And look, I hope it's in this administration. I hope it's in sometime soon that more people can get it. Nothing would make me happier than to see, more kids and more adults who need this, be able to just go to an infusion center, and get this locally.
Yeah, absolutely, man. Well, thank you for the compliment, and congrats on your deal. I hear it's going really well.
Yeah, no, things are great, and I'm grateful for it, but it wasn't easy. So, and the bad actors are tough. So I'm just going to say that thank you, as somebody who has seen the fruits of your labor. You can't necessarily talk specifically about the indication, but I did another episode on autism, here, and it was an inflammatory, response with my son, and within 2 months, it's a remarkable change.
I wrote a paper in 2007 on why I thought stem cells would be useful for the treatment of autism. And I already touched on the anti-inflammatory activity, because I know in a lot of kids with autism, there's too much inflammation in the whole body, too much, particularly in the brain. And also there are certain, in other, not all kids, but some kids, there decreased blood flow to certain parts of the brain. And basically I wrote just very basic. Okay, these cells decrease inflammation. These cells can induce new blood vessel growth. Therefore, maybe they could help autism. And I don't know if you know how many times an average scientific paper is read, but if you take all of them and I think the number of reads is like 3.
And it's usually just your friends. Yeah.
And that's usually you, your wife,
and the professor that taught you about the subject in the first place. Yeah.
right?
Yeah.
I haven't looked at this for a while, but the last time I looked at it, this article was open access, and it's been downloaded over 95,000 times.
I was one of them, so it was 95,001. So yeah, it was a 2007 paper. I remember it's one of the most downloaded medical articles of all time, if not the most, if I recall. I remember reading in your book, and I may get the stat wrong, that by the time we're fully grown, we have something like 10% of the MSCs we had at birth.
Yeah, that's in the bone marrow, which would be your predominant reserve. Yeah, by the time you reach skeletal maturity, roughly 10% are left.
I feel like we're almost describing why we age biologically.
Yeah. Another really interesting thing, I think this is in my book, but if you take postnatal cell that divides every 24 hours, and then you let it divide in culture, just give it nutrition, give it the warm, fuzzy environment that it likes, it will divide every 24 hours. The net result on that is at the end of a month, you'll have 1 billion of those cells. And then if you take a 35-year-old bone marrow, MSC, and plate it, and it divides roughly every 48 hours, every 2 days. No, I think it was 65-year-old, it took 60 hours, and then that number is a paltry few thousand. It's a logarithmic difference if you have highly functioning cells, and that's one of the things that the cells do when they secrete things. They stimulate old cells to behave younger. So you could take old MSCs that are all burnt out, put in young, healthy MSCs, and the old ones, you can separate them by a membrane, so they're not even touching. You can look at the mitochondria, like in an old raggy cell, the cells are all flat, and what remaining mitochondria they're all huddled up around the nucleus, just trying to survive. And then you basically put young, healthy cells and let the secretions of that go in there, and their morphology completely changed. They look younger. They look like 20-year-old cells instead of 80-year-old cells.
I am unbelievably heartened to hear that they are being more receptive and sounds like being more progressive. And they're asking you for help at this point, for guidance, aren't they?
Well, there are some senior HHS officials that we've been talking to who are, by every indication, very interested in making MSCs available, only cells that have been produced in a way where there's definite safety with oversight from FDA, but making them available to a much larger percent of the population than they are now at maybe an earlier stage. This is the only medication I think that I've ever seen that doesn't have serious adverse events. There's a working group of us from industry and from academia who have been talking with some of these advisors and just giving them our thoughts. Nothing's binding. There's nothing official, but we have basically a think tank. How can we very responsibly and safely get cells into the hands of doctors so that they can treat their patients in as efficient way as possible? But number one is safety. And number two is availability. A few points here. One is on cost. The cost of these cells is kind of extraordinary, but I think I've given you a flavor of why they cost so much.
Yeah.
But, we have one facility cost. So let's say I'm burning $1 million a month keeping my facility open, which is not far from the truth. Then if I make one dose of cells, it costs me $1 million. As we produce more cells, more cell lots, the price goes down dramatically. So I think one thing that's going to happen to see a pretty good price decrease across the board, even from the most expensive places, because once you get critical mass and all the overhead is paid for, then the incremental cost is relatively low compared to just covering this massive load that you have of keeping your facility open. That's one thing. The other thing is, look, these cells are thrown in with CAR T cells, which are been a major discovery and a major pathway forward for certain blood cancers and hopefully solid tumors in the future. A typical CAR T cell therapy costs $500,000 and it's kill you. Well, that's regulated in the same bag as basically the same group of people as MSC. Gene therapy, which most recently has resulted in 3 kids with Duchenne muscular dystrophy dying, is in the same category. Cell, tissue, and gene therapy is all in one place at FDA. So maybe there could be a working group at FDA that would be solely schooled and cover MSCs only, because they're in a class by themselves when it comes to potential downside and demonstrated safety profile. There's nothing else in the category, I think, that shares that same safety profile. And it would be great if you didn't have a room of experts that's spending their time worrying about gene therapy, which is so different They're worrying about vaccine manufacturers, which is, all these things together. But just if the cell and gene therapy, every other class of biologic in there in cell and gene therapy is arguably way more toxic than MSCs could ever be. So that's one recommendation. Another one is that there's a very nice model in Japan that has resulted in a lot of innovation. And that's one thing we haven't touched on, but the current system completely stifles innovation.
Mhm.
If it costs you 2 months to get an answer back from the FDA on when you could talk to them in 2 months, and every interaction is costly. I think there's recent testimony by one of the developers of CAR T cell therapy saying we are getting killed by places like Germany where they have hospital exemption, meaning there is a hospital with an institutional review board that says yes or no if they can make a small procedural change based on risk factor. And they're very smart people, and they can make those decisions in a week without having this big layer. They already know the relative safety profile of CAR T cells. They know what's bad and what's good. And to make a minor procedural change to be set back 6 months is stifling for innovation. China has the same thing. They have hospital-based approvals and IRBs that can approve these minor changes. Something as simple, and I've talked about, we did vitamin C work in the past. If you add vitamin C to the culture of MSCs, which has some really interesting potential benefits of making them more anti-inflammatory than without adding extra vitamin C. I can't do that. It would take me 6 months and cost me at least $5 million to do that. Whereas if you're in a hospital-based IRB system, you say, look, we just want to make this change. It's just vitamin C, and it's going to be this dose, which is completely tolerated by human beings. And if it doesn't affect the cells and enhances their ability to help somebody, why not? Just common sense. So there's the Japanese law, which it's a regenerative medicine law that covers cell-based therapies, and it went into effect in 2015. And basically, you demonstrate safety for your cell product, and then you're able to market it for 7 years, so long as there's a registry that they have oversight over. And at the end of 7 years, then you can report to the agency that these are your efficacy. The agency the whole time is looking over safety. But at the end of 7 years, you then either get approval or you're done. But you've had that period of time of good oversight of a product that's already been demonstrated to be safe with a registry, and that we already have the right to try law. It has responsible ways of manufacturing baked into it. If we could have a marriage somehow of our federal right to try law, the Japanese regenerative medicine law of 2015, then I think the industry would survive, thrive, and we could help so many more patients without burning through many. Time is the only thing we have. And when you have to wait 6 months on an answer for something very simple, I think it stifles innovation. It stifles iterations. Look at Elon Musk. Nobody iterates more than Elon Musk, but look what he's done. We're stuck iterating at stuck in molasses.
It's painful, and I'm gonna actually give my own non-binding recommendation to that working group in a second. But I'm actually now looking outside the U.S. for starting our clinical trials. And it's because other countries, Australia is a great example, and there's huge multinational contract research organizations that run these trials, including in the U.S., by the way, that say, hey, if you want to get started, it's a 43% tax rebate if you go in Australia. It takes 6 weeks for the government to approve your protocol, provided you've demonstrated the product is safe. It's not like they're not reviewing the material. It's like I don't want to say they're compensating. That's the wrong choice of word. They're incentivizing research. They're incentivizing development. UK is another great example. They've got like NHRA, I think is the acronym.
MHRA, yeah.
Yeah. So it's this, government working group it's like a business development group for for medical products.
How novel.
I mean, it's fascinating. It's like they actually want to invest in medical technology that could help their population. So you go to them and you say, hey, I've got this idea. They, on their own, will put together a working group of doctors. They bring in their experts on the phone, and then they'll help you run them. They'll help you with the protocols. They'll help you get the centers connected because the socialized medicine makes it easy with, all the different sites out there. They're investing in their populations going forward. And maybe that's just a mechanism because the government pays for all healthcare. I mean, maybe there's a much bigger, theme going there. Like UK has a CEO of their health system. I mean, can you imagine the FDA having a CEO actually forecasting future costs of the population? We're giggling about it, but it may not be a bad thing to think about. It's like if something is going to cost the government, I mean, and Medicare is a huge insurance agency. If something's going to cost the government trillions of dollars or billions of dollars over the course of the next 10, 20 years, maybe we should invest in it now. That's how other countries are thinking.
That's super exciting that an agency is actively trying to help.
They're partnering. Yeah.
Yeah.
Like they want to partner with you. They want innovation. And we're losing products to overseas. We lost, your clinics in Panama. It's not like you wouldn't have put it here if you had the opportunity.
And Australia, we actually went to Australia. We set up a company in Australia because they have that rebate. So it cost me 43% less to do the research there. And our problem was with muscular dystrophy. It's such a small population that there are only 23 million people in Australia, and there were just weren't enough patients to go around. And there are only a few researchers that do that stuff. And then they're basically booked up with drugs. So that's why we didn't go to Australia. But we did go to Australia. We set up a company. The intent was to do our early studies there just because of the cost benefit. But unfortunately, there wasn't a big enough population. But you're right. Every CRO I went to said, don't do this in the U.S., go to Australia,
And it's also twice as expensive here. Even if we took the exact same amount of time, the cost of the sites, the cost of the patients, the cost of everything, just on an average, I believe is about twice as expensive, even if we were just going on the same uniform amount of time. So there's this sort of America first thing going on right now. It's not happening in biotech at the moment. We're losing pharma and biotech because we are not partnering with innovators, and research is a huge business. So, and innovation is important. And if the only thing we're getting is a new wrinkle drug every 15 years, or more like every 15 minutes, it feels like sometimes, I don't think we're investing long-term in the well-being and health of our population. But on the non-binding working group, when you were talking about it, you were talking about the way Japan runs it. It reminds me of something the FDA has already done, actually, but in a different area. So you're in medical, so you know what a compounded medicine is.
Yeah.
So for the listeners, if you go to the doctor and there is no drug that's available that, suits your condition, they are allowed to specialty order from a pharmacy. They can mix things together, a concoction, like a recipe, 2-3 things together that essentially alter an FDA-approved drug. And as a specialty medicine for you, it's an order-by-order basis. Now, back in, I want to say like the mid-2000s, and right around that 2010 area, there were a lot of bad actors in compounding pharmacy. And this is supposed to be a one-by-one basis thing. But they were like mass manufacturing things like pain creams and scar creams and stuff like that, and charging huge amounts of money for it, and essentially marketing them like they were products when they're supposed to be on a one-by-one basis. So FDA, of course, did go in and regulate with as much as they could. I mean, there were a lot of these. The other thing they did is that they actually created a system where you could do it legally, with visibility, and register these products and actually mass manufacture compounds, which is, of course, almost sounds like an oxymoron because a compound is supposed to be a one-by-one thing. That they created this addition to the pharmacy laws that it was called the 503B. 503A is the regular pharmacy. The 503B is this called Compound Outsourcing Center. And they basically allowed these facilities to register with FDA, so they become federally regulated, not just state regulated like normal pharmacies. And then these facilities could mass manufacture compounded drugs, for indications that were being like chemotherapy as an example, saline bags, things like that. And instead of doing it on a one-by-one basis, they could just make a big batch of them. And FDA regulated them, so they were registered with them, but they got the right to do it. And I feel like there may be an interesting opportunity to copy paste sort of what they did for the compounding pharmacies here, because they They had to be GMP, they had to be inspected, they had to have all their quality systems in place in order to sell the drugs. You had to have all the same testing, the aging data, the sterility data, that they were registered and visible. So it sounds a lot like the Japanese model. So my recommendation is they take the 503B thing for compounding and smack it on MSCs. And we don't have to reinvent the wheel if it's already there. We can just kind of drag the cursor in the system. I think that'd be cool. So
You sent me some stuff that was very useful, and I forwarded on them. I think everyone was stuck it.
I would love that. So we can end with me saying that I'm you're one of my science heroes. So it's a huge honor to have you on here. You're one of my family's heroes at the moment because of my son doing so much better since going to your clinic. And I just want to say thank you. And I hope you got a chance to talk about the things you wanted to talk about. I hope that the listeners got a chance to hear more about the MSCs, but there's some serious science here. This isn't just some outside the area witchcraft. Like there's hardcore data backing what you do and years of it. We're just behind here.
Well, Bradley, thank you so much for having me on. It was an honor, pleasure, and I hope your listeners got something out of it. And look forward to talking to you real soon, man. You're one fun guy. You have so many ideas, and your brain works differently than most folks, and I really appreciate that.
I could say the same about you. So between the two of us, you never know what's going to happen. All right.
Right.
All right. Thanks, everybody.